Good morning, biology class. We are again looking at genetics. Wanted to give a quick rundown again of what the study of genetics was, or sorry, what genetics was, and move into some Gregor Mendel back history, and then we'll push ahead and we'll talk about his experiments a little bit. So some of this we've talked about already. Some of this we need to get into. So just as a reminder, how are traits passed on through sexual reproduction? This is really the study of how characteristics are transmitted. That's what inheritance is. So genetics is the study of how characteristics are passed on. So inheritance is how the characteristics are transmitted. Genetics is the study of that. Okay, so genetics in reality is the study of inheritance. Okay, as a reminder, inheritance was really first extensively studied, or is credited to Gregor Mendel, father of modern genetics. He's first to study inheritance with a scientific emphasis. He was actually a physics student at one point, so he was the first one that really used inheritance and put numbers to it. Okay, so he studied essentially heredity, which is transmission of characteristics from parents to offspring. What he focused on was characteristics, so those inheritable or heritable features such as hair color, flower color, plant height, hand size, those types of things, all characteristics. Now, what he decided and what he observed is that, say, for hair color, there was different variations: red, blonde, black, brown, whatever. Now, what he called those are traits, or they're variations of a particular characteristic, and specifically, we'll get into this in our new section that's called an allele. Right? So here's examples of characteristics: hair color, eye color, height. Okay, hair color again has different traits: blonde, brunette, redhead, of course, in this picture. So eye color also has different traits. There's 4 different traits listed here: blue, brown, hazel, green. All of them are variations. Some of those fall into the idea of polygenetic inheritance, which is another step above what we're talking about right now, talking about how many genes are a part of a certain characteristic. Polygenetic inheritance basically states that there are multiple genes affecting eye color and hair color and whatnot. So anyway, that's a lecture for another day. So here's another example. Here's the characteristic: height. Here are the traits: 5 foot, 5'1", 5'2". Here's a kind of a nice picture of apparently a college put together. Here's the shortest people over here and the tallest people right over here. Okay, so now what we're going to get into is, just so now that we've kind of established what a characteristic is and what a trait is, we're going to talk about Mendel's experiments. And I apologize if this is misspelled, but it should be Mendel's experiments. Okay, so this is a view into the world of inheritance. Mendel started, well, first of all, when he first got to where he was studying, he was actually studying bees. Unfortunately, he was breeding bees that became a little too aggressive. Okay, so he had to stop. And then he went to mice. Well, again, something that we don't want more of is mice. So eventually he landed on the humble pea plant. And the nice thing about pea plants, they matured very quickly. They had very distinct traits, and they were very easy to study because of that. So he wanted something that could reproduce quickly in his lifetime, and he could see multiple traits or characteristics easily. Okay. So Mendel developed the hypothesis called the particulate hypothesis. In this case, it states that parents pass on to their offspring separate and distinct factors, what are called alleles, and we'll define that here in just a minute, okay, that are responsible for inherited traits. So really what it looks like, if you want to think about it visually, our genes are more like marbles, not paint. At one point, scientists really thought that genetics was just blending. Okay, so if you think about taking black paint and white paint, you would kind of get a grayish color. That's what they thought genetics was. Mendel observed that it was not necessarily a blending. He observed that, or thought it was going to be more of a marble. These 2 sets of marbles fit together, and one of them will express a trait and one of them won't. So again, here's an allele. An allele is one of the alternative forms of a gene that governs a characteristic such as hair color. The alleles for such a characteristic could be brown hair, black hair, red hair, blonde hair. So that sounds very similar to the term trait. So these are the different factors they talked about. Alleles is what he was discussing. So these are his crosses. Now this is essentially what we're going to talk about. This comes directly out of your book. Okay. On your left, you see the characteristics. There are just a few here. Okay, I did crop this picture. It doesn't have all of them on there. So position of the flowers along the stem. We have axial and terminal. Height of plant. We have tall and short. Pod appearance. It's inflated or constricted. And of course, pod color. You have green and yellow. Right. Next to that column, we have a column for the P generation. This is referring to the parents. These were the two parents that he crossed. Okay, so he had in this case, he had an axial flower and a terminal flower. He had a tall and a short. He had an inflated and a constricted. He had a green and a yellow. Okay, so this was the parent generation. And in the parent generation, we move next to the parent generation, is the F1 generation. This is the offspring. This row right here, these are the offspring that came from these parents over here. So the F1 generation is the offspring of the P generation. Right next to that, you have the F2 generation, which are the offspring from the crossing of two F1 generation individuals. Okay. Then next to that, we have the observed ratios. Here's the more scientific definition right here. And this would be the simplified predicted ratio that Mendel believed would be there. So we'll talk about this a little bit more. First that we need to talk about in discussing his experiments is the idea of true breeding in the P generation. So in the P generation, both the inflated and constricted parents were true breeding. Well, what does that mean? True breeding really talks about purebred. If you've ever heard the term purebred dog or a purebred horse, and those characteristics are important, we need them to come out of there. They're really discussing of this characteristic such as muscle tone in the horse or stride in the horse. They feel some of those things are genetic. Those are characteristics, and they want a specific trait that comes out of there. They want good muscling to come out of there, and we talked about that in cattle as well. We want in the straightness of the back. We want straight back. We don't want a lot of curve to that cow. We want a nice solid frame to add muscle to in the beef industry. Okay, so in the parent generation, in his experiments, he wanted true breeding. He wanted purebred. Okay, so what purebred means is it always produces offspring with the same traits as the parent. So in this case, inflated, this parent here, this inflated pod appearance, that parent would always produce offspring that had inflated pods. This constricted parent would always produce offspring that had constricted offspring. Okay, so these two individuals were purebred. They were always producing inflated, or they were always producing constricted. And same down here, always producing green, always producing yellow. All right, so that's kind of where he had to start. Now, when he crossed these two individuals in the P generation, okay, what he ended up getting, get this out here. What he ended up getting when he crossed these when this cross occurred in the F1 generation, in the offspring, they all turned out to be inflated. We found this to be curious. Same thing with pod color. They all turned out to be green. Okay, so in the F1 generation, after he had true breeding parents that were crossed, in the F1 generation, he had individuals that showed all of one characteristic. So only one trait or one allele was evident in that F1 generation from true breeding parents. That's an important note in his experiments. When he found true breeding individuals and then he crossed two different alleles of those true breeding parents, he only got one. So the other allele in that cross failed to appear. So this is what his conclusion was initially. The trait appearing in the F1 generation was controlled by a dominant factor. So this is where we get the terms dominant and recessive. Okay, so dominant factors masked other traits. Okay, now what he ended up doing then was crossing two of the F1 generations. So he crossed two plants from this generation, showed the inflated pod appearance. So he crossed two individuals. What happened was he got inflated pods, but he also saw the recessive trait, or what we're going to call a recessive trait. He saw the masked trait come out again. And it turns out, as you can see in the ratio here, it turns out to be 3-to-1. So this is going to lead into a few more things when we get into test crosses and whatnot. But essentially, he found out that we have dominant traits and we have recessive traits. Okay, so he sharpened his conclusion. He kept with the idea of a dominant factor, but then he added this. The trait that reappeared in the F2 generation was controlled by a recessive factor, meaning that only if it was paired with another recessive factor. In our little experiment, we talked about this being homozygous recessive. Only when it was paired with the other recessive factor could it appear. So, dominant factors always masked the other trait. Two recessive factors were able to come through, and that occurred in the F2 generation. Okay. The law of segregation was his next conclusion. So he came up with the idea, okay, we have dominant factors and we have recessive factors. After that, he noticed and he stated that a pair of factors is segregated or separated during the formation of gametes. Well, we've talked about that already. We've talked about meiosis. Gametes produced, receive half the number of chromosomes from the parent. That occurs in A1. Okay, anaphase 1 during meiosis 1. We have those homologous chromosomes separating from each other. That happens during A1 and T1, and we get into meiosis 2 and we start separating out chromatids. Okay, but the law of segregation is that these chromosomes will actually separate. Okay, that happens during meiosis 1. He went further and he stated that fertilization is when these two different factors from two different gametes will come back together. So, two gametes combined during fertilization. The offspring have two factors of each characteristic. Again, gives us diversity, and it tells us what our inheritance is going to end up being. Again, when we go to the dominant and recessive, one of those alleles could be dominant and could mask that recessive allele that comes through in the other side. His last statement was a law of independent assortment, which basically states what we've already known. Okay, we talked about this, like I said, in the last section. It states that factors separate independently of one another during formation of gametes. What he did was he studied the crossing of plants that had two different characteristics. Let's just say plant height and flower color. Okay. When he crossed those two, he found that dominant factors from each characteristic did not appear together. What that's basically saying, let's just say plant height, tall plants are dominant, and flower color, purple color is dominant. When he crossbred individuals that had those two different traits, he noticed that a plant could have tall height dominant, but then it could also have recessive color, let's say white. At the same token, he could have a short plant, it's recessive, and he could have a dominant purple color. Because of this change, because of this difference, he stated that apparently these chromosomes do not travel together. Dominant chromosomes or dominant alleles do not always travel together. So it's not going to be tall plants with purple flowers all the time. It's not going to be a short plant with white flowers all the time. They travel independent of each other. So this is what the idea of independent assortment is. You have alleles that travel independently. They separate during anaphase 1. They line up differently during metaphase 1. All those things work in there. So Mendel's experiments were able to look at the idea that there are dominant and recessive alleles. They were able to help figure out the idea of law of segregation, that factors will actually separate, or chromosomes will actually separate during meiosis when we create these gametes. And he was also able to conclude that chromosomes, or more importantly, alleles, independently assorted. They were not combined together beforehand. Dominant did not always travel with dominant. Recessive did not always travel with recessive. They were independent of each other. Okay, so when we come back, we'll talk about the principles of segregation. We'll get into crosses, which is a lot of fun. We'll talk about Punnett squares. We'll talk about test crosses. We'll talk about blue chickens and how you can get your figure out what your blood type is. All right, thank you very much.